The previous video covered one wavelength of sunlight, the ultraviolet B photon that breaks a bond in your skin and starts the vitamin D cascade. 200 genes regulated by a single photochemical event. But sunlight is not one wavelength. It is a spectrum, stretching from ultraviolet through visible light into infrared, and different parts of that spectrum reach different depths in your body and do completely different things when they arrive. The UV story is the one that gets told. The infrared story, the half of sunlight that passes through your skin entirely, penetrates several centimeters into your tissue, and reaches the mitochondria inside your cells, has almost never been told. What happens there is stranger, more direct, and more immediately physical than anything a hormone cascade can do. There is an enzyme inside your mitochondria that absorbs red and near-infrared light and runs faster when the light hits it. And there is a molecule, nitric oxide, bound to that enzyme, blocking it, that the light kicks free. What happens next involves your blood vessels, your joints, and a glucose pathway that has nothing to do with insulin. The skin you think of as a barrier is a solar interface, reading the spectrum and responding in photochemistry. Roughly half the energy in sunlight arrives as near-infrared radiation. Wavelengths between 700 and 1400 nanometers, invisible to the eye, carrying less energy per photon than ultraviolet, but penetrating far deeper into tissue. The physics of penetration depth is governed by the optical properties of biological tissue, specifically by how strongly the tissue absorbs and scatters light at each wavelength. Ultraviolet light is absorbed almost entirely within the first fraction of a millimeter, the epidermis and apodermis. Hemoglobin, melanin, and water all absorb strongly in the UV range, which is why UV cannot reach deeper structures. Visible light penetrates further, a few millimeters, because absorption by those same chromophores is weaker across the visible spectrum. Near-infrared light enters what physicists call the optical window of biological tissue, a range of wavelengths where absorption by water, hemoglobin, and melanin is at a collective minimum. The tissue becomes relatively transparent. Near-infrared photons pass through skin, through the subcutaneous fat layer, and into the muscle and connective tissue beneath, penetrating several centimeters, reaching blood vessels, reaching joint capsules, reaching every cell in the tissue volume the light traverses. Inside each of those cells, the photons reach the mitochondria, and inside the mitochondria, they reach a specific molecular target, cytochrome C oxidase, the terminal enzyme in the electron transport chain. Consider what is happening inside a single cell right now as you watch this. The electron transport chain is the final stage of cellular respiration, the process by which the cell converts the chemical energy in food into ATP, the molecule that powers virtually every energy requiring process in biology. The chain consists of 4 protein complexes embedded in the inner mitochondrial membrane, passing electrons from one to the next in a sequence that pumps hydrogen ions across the membrane and creates the electrochemical gradient that drives ATP synthase. The rotary molecular motor that assembles ATP from ADP and phosphate. Every cell in your body contains hundreds to thousands of mitochondria. Every mitochondrion contains multiple copies of the electron transport chain. The total number of ATP synthase motors running in your body at any given moment is in the trillions, each one a nanoscale turbine, spinning at roughly 100 revolutions per second, assembling 3 ATP molecules per revolution. Cytochrome C oxidase is complex IV, the final enzyme in the chain, the one that accepts electrons at the end of the sequence and transfers them to molecular oxygen, producing water. Without complex IV, the entire chain stalls. The electrons have nowhere to go. ATP production halts. Cytochrome C oxidase contains copper and iron centers that absorb light at specific wavelengths, with absorption peaks at approximately 660 nanometers in the red range and 830 nanometers in the near-infrared. The absorption spectrum of this single enzyme, mapped by Tina Carroll at the Russian Academy of Sciences through painstaking spectroscopy over a career, matches the wavelengths of sunlight that penetrate deepest into tissue. The peaks correspond to specific quantum mechanical transitions in the copper and ion centers. The photon energy at 660 nanometers matches the energy gap between two oxidation states of the copper center, and the 830 nanometer peak matches a transition in the ion-copper binuclear center. When the photon is absorbed, the metal center changes oxidation state, and the conformational shift that follows increases the enzyme's electron transfer rate. Why the match? Because the enzyme evolved in organisms exposed to sunlight, and the metal centers that perform the catalytic chemistry have electronic transitions in the wavelength range that the optical window of tissue transmits most efficiently. The physics of tissue optics and the physics of enzyme photochemistry overlap at exactly the wavelengths the sun provides, a convergence that reflects 2 billion years of evolution under a star that outputs a specific spectrum. The enzyme was shaped by the light. The light reaches the enzyme because the tissue transmits it. The match is architectural. Sunlight contains both wavelengths. When near-infrared photons are absorbed by cytochrome C oxidase, the enzyme's catalytic rate increases. The electron transport chain runs faster. More electrons are transferred to oxygen per unit time. More hydrogen ions are pumped across the membrane. ATP synthase spins faster. The cell produces more ATP, more energy available for repair, for signaling, for protein synthesis, for immune function, for every process the cell performs. The increase is measurable. Cells illuminated with red and near-infrared light at the correct wavelengths show ATP output increases of 15 to 40% in laboratory studies, depending on the cell type and the dose. 15% more energy in a cell that has been energy limited is the difference between a repair process that stalls and one that completes. Something distinguishes the warmth of sunlight from the warmth of a heated room, and the distinction is not psychological. The specific quality of warmth that sunlight creates, deeper than the surface heat of a radiator, felt in the muscle and the joint rather than just on the skin, is near-infrared radiation reaching tissue several centimeters below the surface and stimulating the mitochondria in every cell it passes through. A heated room warms the surface through convection and conduction. The air transfers thermal energy to the outer layer of skin, which conducts it inward slowly, heating tissue by raising the average kinetic energy of nearby molecules. Sunlight energizes the interior through photon absorption, electromagnetic radiation penetrating directly to mitochondria and increasing their catalytic output through a quantum mechanical interaction with a specific enzyme. The mechanisms are entirely different. The body distinguishes between them because they are, in physical terms, different events generating different cellular outcomes. You have felt this. The tiredness that eases when you step outside into full sun on a cold morning. Before the vitamin D has been synthesized, before any hormonal cascade has had time to operate, is partly the mitochondria in your skin and muscle tissue responding to the near-infrared half of the spectrum. The increased ATP is available within minutes. The effect is local and direct. Photon strikes enzyme accelerates, ATP output increases. No intermediary, no signaling cascade. Light energy converted to chemical energy inside the cell through an electron transport chain that has been running in every aerobic organism for roughly 2 billion years. Now, the nitric oxide. When cytochrome C oxidase absorbs near-infrared photons and its catalytic rate increases, a second event occurs simultaneously. Nitric oxide, a small reactive gas molecule, is bound to the enzyme at the same site where oxygen normally binds. In low oxygen conditions or under cellular stress, nitric oxide occupies the oxygen binding site on complex IV and inhibits the enzyme, slowing the electron transport chain and reducing ATP production. This is a regulatory mechanism. The cell uses nitric oxide binding to modulate its own energy output in response to local conditions. When near-infrared light is absorbed by the enzyme, the photon energy displaces the bound nitric oxide. The bond between the nitric oxide and the ion center of the enzyme is broken by the absorbed photon, a photolysis event, the class of reaction that breaks chemical bonds throughout photochemistry. the nitric oxide is released. Two things happen at once. The oxygen binding site is cleared, allowing the enzyme to resume full catalytic activity, and the freed nitric oxide diffuses out of the mitochondria, out of the cell, and into the surrounding tissue. Nitric oxide in tissue is a vasodilator. It relaxes the smooth muscle in blood vessel walls, increasing the diameter of the vessel and improving local blood flow. The mechanism is well established. Nitric oxide activates soluble guanylate cyclase, which produces cyclic GMP, which triggers smooth muscle relaxation. One photochemical event, the absorption of a near-infrared photon by cytochrome, Oxidase, simultaneously restores full enzyme activity, increases ATP production, and improves local blood flow through nitric oxide release. The cell gets more energy and more oxygen from a single photon absorption. The elegance is in the coupling. A single molecular event that accelerates the enzyme also removes the molecule that was inhibiting it, and the released inhibitor becomes a vasodilator that brings more oxygen to the tissue the enzyme is working in. One photon. Three consequences. The physics is economical in a way that engineered systems rarely achieve. This is the basis of a clinical field called photobiomodulation, the use of specific wavelengths of red and near-infrared light to modulate cellular function. Hundreds of clinical trials document measurable effects: accelerated wound healing in diabetic ulcers, shortened post-surgical recovery, reduced inflammation across multiple conditions. The mechanism in every case traces back to one molecular event: photon absorbed by cytochrome C oxidase, enzyme accelerated, ATP output increased, nitric oxide released, blood flow improved. Where this mechanism becomes personal is joint pain. Joint pain. The specific morning stiffness that arrives with age, the knees and hips and hands that take an hour to loosen after a night's sleep, has a measurable relationship with mitochondrial function and local blood flow in the synovial tissue that lines the joint capsule. Synovial tissue is metabolically active. It manufactures the synovial fluid that lubricates the joint, maintains the cartilage surface, and clears the inflammatory debris that accumulates during normal use. All of this requires ATP. All of this requires blood flow. Near-infrared light reaching those joints stimulates the mitochondria in the synovial cells, increases local ATP output, releases nitric oxide that dilates the blood vessels serving the joint, and improves the oxygen and nutrient delivery that the tissue requires for repair and maintenance. The person who has felt their joints ease after 20 minutes in morning sunlight, a loosening that a heated room does not replicate, was experiencing photobiomodulation. The light was doing something the heat could not, reaching the mitochondria directly and changing the biochemistry of the joint tissue from the inside. Mitochondrial melatonin arrived latest in the research, and once I understood it, the rest of the picture looked different. Melatonin is known as the sleep hormone, produced by the pineal gland at night, suppressed by blue light in the morning, governing the circadian rhythm. That is the pineal melatonin story. It is real, well documented, and covered in the previous video. But there is a second melatonin story that has emerged from research over the past 2 decades, and it has almost nothing to do with sleep. Mitochondria produce their own melatonin, locally, inside the cell, in response to stimulation by red and near-infrared light. This mitochondrial melatonin is not released into the bloodstream in meaningful quantities. It stays inside the cell, specifically inside the mitochondria, and it functions as a targeted antioxidant, neutralizing the reactive oxygen species generated as inevitable byproducts of ATP production. The chemistry is specific. The electron transport chain, while transferring electrons to oxygen, occasionally transfers an electron to an oxygen molecule prematurely, before it reaches complex 4, creating superoxide radicals. These are oxygen molecules carrying an extra electron, chemically aggressive, capable of stripping electrons from nearby molecules and damaging whatever they contact. Superoxide can oxidize the lipids in the mitochondrial membrane, altering its permeability and disrupting the electrochemical gradient that drives ATP synthesis. It can damage mitochondrial DNA, which, unlike nuclear DNA, has minimal repair mechanisms, and sits in direct proximity to the electron transport chain that generates the radicals. It can oxidize the iron and copper centers of the electron transport chain proteins themselves, degrading the machinery that powers the cell. This is oxidative stress, and it is cumulative. Across a lifetime, the accumulated damage to mitochondrial DNA, membranes, and proteins reduces mitochondrial efficiency. Cells generate less ATP. Tissues function with less energy. The decline is gradual enough that it feels like aging, like the slow dimming of a system rather than a specific injury, because it is happening in every cell simultaneously, across every tissue, across decades. The person who notices that their recovery from exertion takes longer than it used to, that a night of poor sleep affects them more deeply than it did at 40, that wounds heal more slowly and muscle soreness persists longer, is observing, in part, the downstream consequences of mitochondrial efficiency declining. The cells that repair the tissue have less energy to do the work. The immune cells that clear damaged protein have less ATP to fuel the process. The system still runs. It runs slower, with less reserve, and the margin between adequate function and impaired function narrows with each decade. The research into mitochondrial dysfunction as a driver of age-related disease has grown exponentially over the past 20 years because the connection is so direct. Damaged mitochondria generate less energy and more radicals, which damage the mitochondria further, which reduces energy output further. The cycle accelerates. The body built a countermeasure, and the trigger for that countermeasure is light. Melatonin, produced locally inside the mitochondria in response to red and near-infrared light, scavenges those superoxide radicals before they can damage the structures that created them. The antioxidant is being manufactured at the site of the damage, triggered by the wavelengths that are simultaneously increasing ATP production through cytochrome C oxidase stimulation. The system is self-protecting. The light that accelerates energy output also triggers the synthesis of the molecule that protects the machinery from the damage that accelerated energy output creates. The mitochondrial production pathway was established by Russell Reiter's work at the University of Texas. Years of melatonin biochemistry demonstrating that red and near-infrared wavelengths specifically trigger the synthesis. The melatonin the mitochondria manufacture locally is targeted at the structures that need it, a precision that systemic supplementation cannot replicate. Oral melatonin, the pill sold as a sleep aid and antioxidant, reaches the bloodstream, crosses the blood-brain barrier, and affects pineal-regulated sleep architecture, but it does not concentrate inside the mitochondria of illuminated tissue at the levels that locally synthesized melatonin reaches. The mitochondrial pathway is not systemic hormone delivery, it is a local manufacturing operation, the antioxidant built at the site of the damage, in response to the signal that accompanied the damage for the entire evolutionary history of aerobic life. A person taking an antioxidant supplement while spending their days indoors is supplementing one pathway while leaving the primary local antioxidant system without its trigger signal. The trigger is sunlight. The response is local, the protection is precise. Reiter's finding reframes the relationship between sunlight and cellular aging. Without the red and near-infrared signal, the mitochondria still run. They still transfer electrons, still generate ATP, still create superoxide as a byproduct, but they run without the antioxidant that the light would have triggered. The damage accumulates faster. The mitochondrial decline that feels like aging is, in part, a consequence of running the electron transport chain without the photochemical signal that was supposed to accompany it. Indoor life does not stop the engine. It removes the maintenance system. The research is still establishing how large this effect is in human tissue at natural sunlight doses, but the direction is consistent across every model tested, from isolated mitochondria to cell cultures to animal studies. The mechanism is real. The magnitude in a living person standing in morning sunlight is what remains to be precisely quantified. One mechanism reaches a different organ entirely. High-lux outdoor light, 10,000 to 100,000 lux, stimulates dopamine release from amacrine cells in the retina, and retinal dopamine regulates the physical elongation of the eyeball during development. Ian Morgan at the Australian National University established that the myopia epidemic, 30% of the global population, projected to reach 50% by 2050, is driven by the shift from outdoor to indoor life. A photon count problem, the retina needs high lux to release enough dopamine to regulate eye growth, and indoors cannot deliver it. If you spent your childhood outdoors, walking to school, playing in fields, spending summers in full sunlight, your retinal dopamine regulated your eye growth across the critical window. Your grandchild, growing up in classrooms lit at 300 lux, receives a fraction of that signal. The eye elongates because nothing in the light environment is telling it to stop. 2 hours of outdoor time per day is enough to significantly reduce the risk. The physics was available to prevent it. The lux was not. One more mechanism. The nitric oxide released by sunlight from the skin. A different UV-driven pathway from the photobiomodulation nitric oxide has a direct effect on blood sugar that operates independently of insulin. UVA light, longer wavelength ultraviolet, 320 to 400 nanometers, penetrates to the dermis and releases nitric oxide from preformed reservoirs in the skin. This is a separate mechanism from the cytochrome C oxidase pathway. UVA photons break nitrogen-oxygen bonds in nitrosothiols and nitrates stored in dermal tissue, releasing free nitric oxide that enters the bloodstream. The cardiovascular effects, vasodilation, blood pressure reduction, were covered in the previous video. The glucose effect was not. Nitric oxide in muscle tissue activates an enzyme called AMP, activated protein kinase, AMPK, which triggers the translocation of GLUT4 glucose transporters to the muscle cell membrane. GLUT4 is the protein that allows glucose to cross the cell membrane and enter the muscle cell from the bloodstream. In a healthy cell, insulin binds to its receptor on the cell surface and initiates a signaling cascade that moves GLUT4 transporters from storage vesicles inside the cell to the membrane, where they open channels for glucose entry. This is how the body clears glucose from the blood after a meal. Insulin tells the muscle cells to absorb it. In insulin-resistant states, the metabolic condition that precedes and defines type 2 diabetes, that signaling cascade is attenuated. The receptor still binds insulin, but fewer GLUT4 transporters reach the membrane. Less glucose enters the cell. Blood sugar remains elevated. Nitric oxide drives GLUT4 translocation through the AMPK route, a completely separate pathway that does not depend on insulin receptor signaling at all. The insulin pathway is impaired. The nitric oxide pathway is intact. Sunlight, by releasing nitric oxide from the skin, is activating glucose uptake in muscle tissue through a route that bypasses the impaired insulin signaling entirely. The two pathways converge on the same outcome, glucose entering the muscle cell, but they arrive there through different molecular doors. Losing one does not close the other. Consider the population-level significance. Type 2 diabetes affects roughly 500 million people worldwide, and the number is rising in every age group above 50. The management paradigm, diet, exercise, medication, monitoring, addresses the insulin pathway and the caloric balance. The nitric oxide pathway has not entered the management conversation because the photobiology research and the endocrinology research exist in separate literatures. A person walking outdoors in sunlight is receiving both the exercise benefit and the photochemical benefit simultaneously. A person walking indoors on a treadmill under LED lighting receives the exercise benefit alone. The difference is the UVA-driven nitric oxide release that the indoor environment cannot provide. Anyone managing blood sugar through diet, through medication, through the daily arithmetic of what they eat and when they walk, has been unaware that the sunlight on their skin during the walk was performing a direct, insulin-independent glucose uptake mechanism in their muscle tissue simultaneously. The walk and the sun were doing more together than either was doing alone. The nitric oxide from the UVA photons was opening a separate pathway into the muscle cells, while the exercise was consuming the glucose those cells were absorbing. A morning walk indoors on a treadmill. Same distance, same speed, same exertion. does not trigger the UVA-driven nitric oxide release. The GLUT4 translocation through the AMPK pathway does not occur. The walk is doing its work. The sun is not doing its work. The two mechanisms, which evolved to operate together in an organism that spent its days outdoors, are being separated by the built environment. This is not an argument to alter medication or abandon dietary management. It is a mechanism that was running measurably, that was never mentioned, and that the person managing their condition was never told about because the research exists in photobiology journals that endocrinologists do not typically read. 6 mechanisms across both videos, each discovered in a different laboratory, published in a different journal, studied by researchers in a different field. None of them knew they were describing parts of a single system. Stand back from the individual mechanisms and the full picture is a system. The skin reading the solar spectrum and responding across every wavelength band simultaneously. UVB photons absorbed in the epidermis break the carbon-carbon bond in 7-dehydrocholesterol and initiate the vitamin D cascade. 200 genes regulated. Calcium metabolism governed. Immune function modulated. UVA photons penetrating to the dermis release stored nitric oxide. Vasodilation, blood pressure reduction, insulin-independent glucose uptake. Red and near-infrared photons passing through the skin entirely are absorbed by cytochrome C oxidase. ATP output increased, bound nitric oxide released locally, blood flow improved. Those same wavelengths trigger mitochondrial melatonin synthesis, targeted antioxidant protection at the site of energy production. High-lux full-spectrum light reaching the retina stimulates dopamine release from amacrine cells, regulating the physical growth of the eye. 5 wavelength bands in the spectrum you stand in every time you step outside. 5 molecular targets generating 5 different compounds the body requires, all running simultaneously in the 15 minutes of morning sunlight that most people no longer receive. The skin that was understood as a barrier, keeping the outside out and the inside in, is an active solar interface, reading the spectrum and converting each wavelength band into the specific photochemistry the body needs. The person standing in morning sunlight does not feel 5 separate mechanisms activating. They feel one thing, a warmth that is different from indoor warmth, an easing in the joints, a lifting of the mood, a clarity in the thinking, an energy that was not there 5 minutes ago. Each of those felt experiences corresponds to a specific photochemical event running in a specific tissue at a specific depth. The warmth is mitochondrial stimulation. The joint easing is local nitric oxide release. The mood lift is the beginning of serotonin and melatonin modulation. The clarity is cortisol regulation. The energy is ATP. Manufactured locally, in the cells the light reaches through an enzyme that has been waiting for the signal since the person went indoors last night. The system took billions of years to assemble. Every aerobic organism on the planet runs some version of the Cytochrome C oxidase pathway. The melatonin antioxidant system predates the evolution of animals. The nitric oxide signaling system is shared across vertebrates and invertebrates alike. These are not recent adaptations. They are foundational biochemistry, built into the deepest layers of cellular machinery, dependent on the one energy source that was always available, the electromagnetic radiation arriving from the nearest star. Modern life has placed most people in environments where this system rarely activates. A standard LED bulb emits 0 near-infrared radiation, no UVB, no UVA. The lux count is 1 to 3 orders of magnitude below what outdoor environments deliver. Indoor lighting allows you to see. It does not contain the wavelengths that any of these 5 systems respond to. The consequences accumulate across decades. In every organ reached by a wavelength that is no longer arriving. Each consequence has been studied independently. The connection, that all of them trace back to the absence of one environmental signal, is the story that has not been told clearly enough. A person who spends 15 minutes in morning sunlight is running all 5 systems simultaneously. A person who spends those 15 minutes under indoor lighting is running none of them. The difference is not incremental. It is the difference between a system that is active and a system that is silent. Tomorrow morning, step outside. 15 minutes. Full spectrum. The machinery is waiting.